Good morning, my dear friends. I hope you all are doing great. So guys, let us start our work. So we'll be with the question number 21. Which one among the following will have 5 chiral centers? A very easy question. The glucose with open structure, the glucose with closed structure, fructose open structure and fructose closed structure. So let us have a look. If we talk of the open structure of a glucose, we will be having 4 chiral centers. Everybody is aware about the second, third, fourth, fifth and the sixth one. CH2OH, OH, H, OH, H, third carbon is OH on left side, H, OH and H. This is the open structure of glucose. So this is open structure of glucose. And how many chiral centers? If we start numbering 1, 2, 3, 4, 5 and 6, we know that the second one, third one, fourth one and fifth one, that means 4 chiral centers are there. Second carbon, third carbon, fourth carbon and fifth carbon. So number of chiral centers. So number of chiral carbons are how many? 4. Okay. So my dear friends, in glucose we will have here M value, the number of chiral carbons of 4. If we look at the closed structure of a glucose, the closed structure of glucose is like this. It should have 5 chiral centers. Have a look. H, OH, H, OH, OH, H, H, OH, H, then CH2OH. This is first carbon, second, third, fourth, fifth and sixth. This is the closed structure of glucose. The closed structure of glucose will have how many chiral centers? The first one. The first carbon is also a chiral carbon here, because here we'll be having H, OH second group, and this is third and this side is fourth. So the first one is chiral, second one is also chiral, third one is also chiral, fourth one is also chiral and fifth one is also chiral. So total 1, 2, 3, 4, 5. Here the N value is number of chiral carbons are 5. So my dear friends, in closed structure of glucose we have 5 chiral carbons. Have a look here. So here the number of chiral carbons are 5. Correct? Now if we talk of fructose and open structure of fructose and closed structure of fructose, we will find out that how many chiral carbons are there as well. So let me take down this thing now. I hope you have written this thing. So let us draw fructose molecule. The fructose molecule will be C double bond O. Here we have CH2OH, first, second, third, fourth, fifth and the last one is sixth. Okay, OH, OH and here we have OH, H, H, H. Look here, this is fifth carbon, fourth carbon, third carbon, second carbon and first carbon. First, second and sixth, they are achiral, they are not chiral. Here we have third carbon as chiral, fourth carbon as chiral and fifth carbon as chiral. So number of chiral carbons are basically here 3. In which molecule? Fructose. Which structure? Open structure. So open structure of fructose will have 3 chiral carbons. So N value is here 3. So let's talk about now fructose in closed structure. Okay. Fructose in closed form. Have a look here, my dear friends. CH2OH, that is 1, 2, 3, 4, 5 and another one is 6, which is outside. H, H, OH, OH and H. If we look at the first carbon, it's not chiral and sixth carbon is not chiral. First and sixth are not chiral. But here the second carbon becomes chiral, third carbon is also chiral, fourth carbon is also chiral and fifth carbon is also chiral. You can check it out. Chiral carbon should be attached to 4 different groups. So if you look at the second, third, fourth and fifth here, they are chiral. So total are 4 chiral carbons here. So my dear friends, here the N value is 4. So dear friends, here N value is 4 in case of Fructose. But which form of fructose? Closed structure of fructose. So our question was, which one among the following will have 5 chiral centers? So 5 chiral centers are present in glucose, but which form of glucose? The closed structure of glucose will have 5 chiral centers, n is equal to 5. I hope you have understood, my dear friends. Please make note of this. Coming to the question number 22nd, which one among the following is a steroid hormone? If you look at the first one, cortisol, cortisol is a steroid hormone. Have a look. It is released by Rrna fasciculata region or layer of adrenal cortex. If we classify the steroid hormones, we have classified steroid hormones into 2 types: adrenal cortex hormones and sex hormones. That means adrenal cortex hormones are the steroid hormones and sex hormones are also steroid hormones. This guy is our adrenal cortex hormone. Okay? So when we say adrenal cortex hormone, so it is obviously a steroid hormone. Steroid hormone. So our answer is the first option itself. So first option is cortisol, and cortisol is our steroid hormone. Now if you come to thyroxine, okay, it is produced by thyroid gland. And you know which kind of hormone is this thyroxine? It's produced by thyroid gland, and it is a amino acid derivative hormone. Amino acid derivative hormone. If you look at insulin, it is produced by beta cells of pancreatic islets, and it is obviously a peptide hormone, or you can also call it as protein or peptide hormone. So my dear friends, our answer is cortisol, and cortisol is produced by the Rrna fasciculata layer of adrenal cortex, and it's a steroid hormone. Correct? So please make note of this. Let's talk about the question number 23. Which one among the following does have deconfiguration? My dear friends, when we talk of capital D, always remember it's related to the arrangement of OH group on the right side of last chiral carbon. Configuration, we can have 2 configurations, D configuration and L configuration in case of sugars, correct? D configuration basically means the presence of OH group on right side of last chiral carbon, correct? So have a look here. If we talk about D configuration, this is capital D. If it is small d means dextro, and that also means the light, plane polarized light, is rotated in clockwise direction. But it is not related to rotation. I have told already in theory classes that this capital D stands for the configuration, like the arrangement of OH group on the right side of last chiral carbon. So D configuration, okay, it represents presence of OH group on right side of last chiral carbon in sugars I am talking about. So here our discussion is about sugars. So if the OH group is present on the right side of last chiral carbon, then it's a D configuration. If it is present on left side of last chiral carbon, then it's a L configuration. So let us have a look. If you look at the first molecule here, okay, you will find out that this central carbon is here chiral carbon, correct? When this is a chiral carbon, let's number it as 1, 2 and 3. We have 3 carbons here, but only the second carbon is chiral carbon. So this second carbon is only one chiral carbon. In first option, we have only one chiral carbon, and that carbon is second carbon, correct? So let us see the position of that OH group on that carbon. Because this second carbon which is here is a chiral carbon. It is only one chiral carbon, so you can also call it as last chiral carbon or first chiral carbon. It's both. Okay? So what is the position of OH group on this last chiral carbon? Remember once again, it is first chiral carbon as well as last chiral carbon, because there is only one chiral carbon in this molecule. So where is the position of OH group on this chiral carbon? Okay? It's on left side. So it's on left side. So when it's on left side, understand that it will have L configuration. Consider this second carbon as first as well as last. You can also call it as last chiral carbon, because there is only one chiral carbon here. Now have a look here. 1, 2, 3, 4, 5 and 6. We have here 2, 3, 4 and 5. So we have here 4 chiral carbons, 2, 3, 4 and 5, correct? Everybody knows about it. So the fifth one is our last chiral carbon here. So the fifth one is our last chiral carbon. And where is the OH group on last chiral carbon? The OH group on fifth carbon, that is last chiral carbon, is on right side. So it's on right side. When it's on right side, and this is our deconfiguration. So my dear friends, our answer is which option? B option is our answer. Let us go to the C option here. 1, 2, 3, 4, 5 and 6. Second carbon is chiral, third carbon is chiral, fourth is chiral, fifth is chiral. Fifth is our last chiral carbon. Fifth carbon is our last chiral carbon. And here the last chiral carbon has OH group on which side? Left side. And when OH group is present on left side of last chiral carbon, we call it as L configuration. So this guy is our L configuration. Now if you look here, it's 1, 2, 3, 4, 5 and 6. Chiral, 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 chiral. Correct? We have chiral carbons here. Fifth is also chiral carbon. So I can call it as last chiral carbon. And it has OH group on left side. When it has OH group on left side, it's again L configuration. So my dear friends, our answer is the B option. Please make note of this. Let us go for the question number 24. Which of the following is required for protein synthesis? My dear friends, we have first of all mRNA, tRNA and rRNA. You know that the mRNA stands for messenger RNA, transfer RNA, ribosomal RNA, and the last option is all of these. Okay, have a look. If we look at mRNA, my dear friends, okay, what is the role of mRNA? It does have an information, okay, in the form of genetic codes for protein synthesis. If you look at tRNA, what is the role of tRNA? tRNA, it transfers, especially there are 20 different types of tRNAs, so it transfers amino acid from cytoplasm to the ribosome for protein synthesis. So the role of tRNA is to carry the amino acids from cytoplasm to the ribosome for which thing? For protein synthesis. Because I told you in theory classes that this tRNA has an anticodon, okay? It will read the information from mRNA that which amino acid is required, correct? So tRNA has an anticodon, okay? It will read the information on mRNA. then it will bring the amino acid from cytoplasm, and there are 20 different tRNAs in the cytoplasm, correct, or in a cell. So once it brings the amino acid from cytoplasm of the cell to the ribosome, then protein synthesis will take place. Those amino acids will be combined on the surface of ribosome through peptide bonds, and protein formation will take place. Then what is the role of this rRNA? rRNA is basically ribosomal RNA, you can say, or ribosomal RNA, you can say. Okay, it is an essential component of ribosome. So it's an essential component of ribosome, because ribosome is a place of protein synthesis. Okay, it's a place of protein synthesis. And you know one thing, this ribosome is made up of two things. It's made up of this rRNA plus proteins. Correct? So my dear friends, all these three things help out in the protein synthesis. So the answer is all of these. I hope this much information is enough. Please make note of this. 25th question, my dear friends. How many hydrogen bonds are formed between nitrogenous bases like adenine and thymine in DNA? It's a very easy thing. If you look at the DNA structure, it's a double helix structure. Everyone is aware about it. So it's from 5 prime to 3 prime, and it's from 3 prime to obviously 5 prime. Correct? Anti-parallel strands. Okay. If we look at, there will be adenine and thymine, which are basically complementary base pairs. So we have here complementary base pairs. Complementary base pairs in DNA. Who are those? Adenine will base pair with thymine. Like if you have adenine here, its opposite base will be thymine. And remember, there will be 2 hydrogen bonds between adenine and thymine. We can call them as complementary base pairs. So these 2 are complementary base pairs, correct? And there is a cytosine, it will be versus guanine. The base pair will be guanine. So between cytosine and guanine in DNA, there will be 3 hydrogen bonds. So guys, here we have 2 hydrogen bonds. And between cytosine and guanine, we will have 3 hydrogen bonds. And these are called as complementary base pairs. Who is the complementary base of adenine? Thymine. Who is the complementary base of cytosine? Guanine. So the answer, the total number of nitrogen bonds between adenine and thymine are 2. 2 is the answer. And 3 is basically the number of hydrogen bonds between cytosine and guanine. Please make note of this. Let's have a look at question number 26. As per Chargaff's rule, what is the ratio of complementary bases in DNA? So we have to find out the ratio between complementary bases, and we know what are the complementary bases in DNA. My dear friends, if you look at, you have complementary bases in DNA like adenine versus thymine and cytosine versus guanine. They make 2 hydrogen bonds, and this cytosine and guanine will make 3 hydrogen bonds, that we know. Adenine will be always equal to thymine. So adenine-thymine ratio, these are 2 complementary bases. Okay, the ratio will be always 1:1. Here also cytosine will be always equal to guanine. So when cytosine equals to guanine, because cytosine will be the always complementary base for guanine, so they will be equal. So here also the ratio between cytosine and guanine will be 1:1. So in DNA molecule, remember, adenine will be always equal to thymine, and this complementary bases like cytosine and guanine, they will be also equal. Cytosine will be equal to guanine. So ratio between these two complementary bases is 1:1, and between these two complementary bases ratio is also 1:1. Hydrogen bonds are 2, hydrogen bonds are 3, correct? So my dear friends, don't compare adenine and cytosine, because adenine and cytosine are not complementary bases. You have to always compare adenine with thymine. Okay, ratio will be 1:1 because adenine will be always equal to thymine in a DNA. And ratio between cytosine and guanine, which are also complementary bases, they will be also equal in DNA, and that means the ratio will be 1:1. Yes, this rule can be violated only when there is a mutation of a particular nitrogenous base. If one nitrogenous base is getting converted into another, let's suppose if thymine gets converted into uracil through mutation, then that ratio 1:1 ratio will be lost. That will be lost only through the mutation process. In general, the ratio between complementary bases will be 1:1. That is the answer for your question. My dear friends, let's look at the question number 27. Match the following and come up with the correct option. So the options are given right out here with you. So you have to match up, okay, these with them. Okay, have a look. If we look at the vitamin A, my dear friends, vitamin A is known as retinol. Okay, and it's very much important for the vision, correct? And if we look at the vitamin B1, vitamin B1 is basically thiamine. Vitamin D is calciferol, and vitamin B2 is riboflavin. So these are the trivial names of these particular vitamins. Okay. Now everybody knows that vitamin A deficiency disease in second column is here. Vitamin A will cause night blindness, and I have also explained you that night blindness basically comes from xerophthalmia. Xerophthalmia is basically hardening of the cornea of an eye. Okay. First of all, xerophthalmia will happen. Then after that, the eye will lose its transparency. Okay. Then after that, it will lead to the night blindness. So remember the term xerophthalmia. That means the first vitamin A, okay, will be matched up with xerophthalmia. So it's like this. So 1 with Q. So the first option if we see here, 1 with Q, that is vitamin A, okay, leads to the deficiency disease called as xerophthalmia. Have a look. This is xerophthalmia here. And what happens in xerophthalmia? Hardening of cornea, or we can say cornea of an eye. Then if you look at vitamin B1, it's a thiamine. It leads to beriberi. So here it belongs. And you know what happens in beriberi? There is a loss of appetite. What is the meaning of loss of appetite? Means you will lose the desire to have food, or you won't feel hungry. Okay? So the loss of appetite will happen. You won't feel like having food. Okay? So that means it's because of the deficiency of vitamin B1, the deficiency disease. If the vitamin B1 in your body becomes less, it will cause beriberi disease, correct? But now let us look at vitamin D. Everybody knows it's calciferol, okay? And vitamin D will cause rickets. And you know that rickets is basically softening of bones, we can say, or deformities in bones of children. It will also lead, vitamin D deficiency will also lead to osteoporosis, like deficiency I am talking about. So vitamin D is very much important for the bones, teeth, correct? So if there is a deficiency of vitamin D, the deficiency disease will be yours rickets, and rickets is mainly happening in the children. But if we talk of the adults, there will be basically the osteoporosis. Okay. Now vitamin B2 is known as riboflavin, and the deficiency of riboflavin will cause a disease known as callosis. And you know what is callosis, my dear friends? Have a look. Let's write down here. Callosis basically leads to the fissures in the corners of mouth and lips. The fissures basically here means cracking. The corners of your mouth will have the fissures in it, cracks in it, and the lips will also get the cracks in it. So it's because of callosis. And the callosis disease is basically a deficiency disease. It's because of the deficiency of vitamin B2. Okay? So my dear friends, deficiency of vitamin A will cause xerophthalmia. Vitamin B1 will cause beriberi. Deficiency of vitamin D will cause rickets in children, and osteoporosis in the adults. And vitamin B2 will cause callosis, and callosis means fissuring in the corners of the mouth and the lips. Now if you look here, first I will match up with Q, I match up with Q, second match up with P, second match up with P, third match up with S, third match up with S, fourth match up with R, fourth match up with R. That means our first option is the correct answer. Please make note of this. Let's have a look at question number 28. How many disulfide bonds are present between two polypeptide chains in insulin? So you have to tell how many disulfide bonds are present between two polypeptide chains in insulin. Between two polypeptide chains we have two disulfide bonds, correct? So let's have a look. If you look at insulin structure, I told you last time, if you remember, that insulin contains 51 amino acids. Okay? And this insulin is our protein, and even it's our protein hormone, we can say. It's a protein hormone. Correct? If I say this insulin, it's made up of two chains actually. Have a look. Its structure is made up of chain A and chain B. If we look at a chain A, it will be made up of 21 amino acids. And the chain B is made up of 30 amino acids. So this chain A is our polypeptide chain. Polypeptide chain is a polypeptide. Chain B is also a polypeptide because it's made up of peptide bonds between amino acids. Between this first and the second, okay, polypeptide chain, between this and this, there are two disulfide bonds. Disulfide bond means SS bond is a disulfide bond. 1 and 2. So we have here two disulfide bonds. So the disulfide bonds between chain A and chain B, or between polypeptide A and polypeptide B, polypeptide B, polypeptide A, these are called as interpolypeptide disulfide bonds. Question tells us how many disulfide bonds are present between two polypeptide chains in insulin. So between polypeptide chain A and B, how many disulfide bonds are here? So we have here, my dear friends, we have here two disulfide bonds. Correct? So answer will be 2. Between two polypeptide chains in insulin, there are two disulfide bonds. And you know one more disulfide bond is also there. Have a look. Okay, this disulfide bond is present in chain A or we can say this disulfide bond is present in polypeptide chain A. But between two polypeptide chains, we have two disulfide bonds, okay, between two polypeptide chains. And this disulfide bond is present in chain A only, okay. So my dear friends, the total number of disulfide bonds will be 3. But between two polypeptide chains, we will have only two disulfide bonds. Now I told you, if you remember, I have told you that these disulfide bonds are formed between cysteine amino acids. Cysteine and cysteine. So cysteine amino acids will make these disulfide bonds. In examination, if they ask you how many disulfide bonds are present between two polypeptide chains, two disulfide bonds. Total number of disulfide bonds in insulin are 3, correct? And these two disulfide bonds which you are looking here between two polypeptide chains, these two disulfide bonds are called as interpolypeptide disulfide bonds. Disulfide bonds. And this disulfide bond is present within chain A. Within chain A only. So this bond is called as intrapolypeptide, means present within the same chain. Intrapolypeptide disulfide bond. I hope you have understood, my dear friends. Between two polypeptide chains with 21 and 30 amino acids, there are two disulfide bonds, and they are interpolypeptide disulfide bonds. Correct? So two interpolypeptide disulfide bonds are present between two polypeptide chains in insulin. Correct? Total number of disulfide bonds are 3. 2 are interpolypeptide and 1 is intrapolypeptide, and that intrapolypeptide is present in chain number A with 21 amino acids. Correct? Please have a look at this. So question number 29. What is an alternate name for glycosidic bond linkage? We have to find it out. So what is the another name of glycosidic bond linkage? My dear friends, have a look. Is it amide linkage, phosphoanhydride linkage, ether linkage, or ester linkage? If we look at the amide linkage is also known as, you can also call it as peptide linkage. So it's your peptide bond. You know that this peptide linkage can be demonstrated like this. Have a look. CH R1 NH2 C double bond O NH CH R2 then COOH. So this is your basically the amide. CONH is your amide linkage, or it's also known as, this is your amide, or you can say peptide linkage. But I have to talk about what is the alternate name for glycosidic bond linkage. So this is not your answer. Amide linkage is also known as peptide linkage, and it is present between amino acids. So amide linkage or peptide linkage is present between amino acids. Now let's have a look at the second one. So first one is not the answer, because amide linkage or peptide linkage is present between amino acids. CONH linkage. So my dear friends, now this amide linkage is nothing but C double bond O and NH linkage. This is your amide linkage or peptide linkage, CONH. Let's talk about phosphoanhydride linkage. You know that phosphoanhydride linkage, last time I told you in ATP molecule. If you look at ATP molecule, you will find out, have a look. You have an adenine attached to the ribose sugar. Then you have phosphate groups, double bond O, O minus, O, P double bond O, O minus, O, P double bond O, O minus, O minus. This is your ATP. And in this ATP molecule, we have this POO and PO bond like this. POO and PO. This is basically our phosphoanhydride linkage. P double bond O, O, P double bond O. This is phosphoanhydride linkage. You can also call it as here P double bond O and P double bond O is your phosphor anhydride linkage. So my dear friends, in ATP molecule between phosphates we have phosphor anhydride linkage. So this linkage will be ours, P double bond O, P double bond O linkage present in ATP molecule, adenosine triphosphate. So after that, let's have a look at ether linkage. Remember my dear friends, this ether linkage is actually known as glycosidic linkage. So your answer is basically C option. How? Have a look. If you talk of this glycosidic linkage, it will be like this. If you have a sugar with you, let's take another sugar molecule, OH, CH2OH, CH2OH. Now this is first carbon and this is the fourth carbon, like 1, 2, 3 and 4. This is alpha D glucose, this is also alpha D glucose. So we have two molecules of alpha D glucose. You can take any glucose molecule, but I am taking here alpha D glucose molecule. This glycosidic bond can be found in between any two sugar molecules, correct? So what will happen? The water loss will be here. OH and H will be lost. OH and H will be lost. So what will remain here? O. So there is a formation of glycosidic bond like this. So if I call this as a carbon here, this is also a carbon here. So it is a COC bond, correct? COC bond, and this COC bond is basically a glycosidic bond. Glycosidic bond. There's a loss of water molecule here, and there's a formation of COC bond. So my dear friends, this glycosidic bond, which is also known as ether bond, is a COC bond. So this is COC bond. And you know, my dear friends, if this glycosidic bond is a COC bond, it can be also represented as ROR bond. And this ROR, this O stands for here ether. That's why it's known as ether linkage also. When you have COC bond is nothing but it's a glycosidic bond or ether linkage. Why it is ether linkage? Because this O between two carbon atoms means these two carbon atoms are two R groups, correct? So between two R groups, when you have an oxygen, that oxygen is known as ether. And so that's why it's also known as ether linkage. So because of the presence of this ether, okay, it will be also considered as now ether linkage. And because this O represents ether, correct? So now we have an ester linkage. Ester linkage will be mainly between carboxylic acid and alcohol. If you have a carboxylic acid with you, OH, and then you have an alcohol with you, let's suppose you have OH, correct? So there is a loss of water molecule here. Due to loss of water molecule, you will have C double bond O. Then what will remain here? O and R. And this COO linkage is basically ester linkage. Okay, so this is the ester linkage. So it is between carboxylic group. When carboxylic group will interact with alcohol, so there is a formation of an ester, and this molecule is ester, and the linkage is ester linkage here. Correct? COO linkage is known as ester linkage. So our answer is this one. Ether linkage is also known as glycosidic linkage. C option is the answer. So dear friends, let's look at the question number 30 here. I have told you last time, if you remember, reducing sugars. Okay, what are reducing sugars? They can reduce Tollens reagent and Fellings solution. Correct? Non-reducing sugars are those which cannot reduce Tollens reagent and Fallings solution. Our question is that which one among the following is not a reducing sugar? That means I have to talk about non-reducing sugars. Correct? So non-reducing sugars will not reduce Tollens reagent and Fallings solution. In Tollens reagent, last time you remember, if you look at the Tollens reagent, I have told you, here we reduce in Tollens reagent, cellular Ag is reduced from +1 to 0 oxidation state. In Fallings solution, copper, copper is reduced from +2 oxidation state to +1. So who will reduce Tollens reagent and Fallings solution? Your sugar molecules will reduce them. And if you understand this factor now here, my dear friends, I told you last time, if you have monosaccharides with you, or you have a disaccharides, and you have, let us suppose, polysaccharides. In polysaccharides you will have glycogen, we can write down here first starch, we have a cellulose, we have glycogen. Okay, so these are all non-reducing sugars. These are all non-reducing. They won't reduce silver from +1 to 0. They won't reduce copper from +2 to +1. So they won't be able to reduce Tollens reagent and Fallings solution. Disaccharides, we have here maltose, lactose, and we have sucrose. These two are reducing, means they can reduce Tollens reagent and Fallings solution, and sucrose is non-reducing. This guy is non-reducing. Correct? Monosaccharides means all monosaccharides are reducing sugars. Are reducing sugars. So my dear friends, glucose is a monosaccharide. So it will be it's our reducing sugar. So it's reducing sugar. Fructose is also a monosaccharide. Correct? It's also a reducing sugar. Because all monosaccharides are reducing sugars. So reducing sugar. If you look at sucrose molecule now here, sucrose molecule is basically a disaccharide, or sucrose molecule is a disaccharide, it's non-reducing. So it's a non-reducing sugar and it's a disaccharide. So remember, sucrose is the only disaccharide which is non-reducing. Other two disaccharides like maltose and lactose are reducing. Maltose is also a disaccharide, but I told you that maltose is reducing sugar. So it is a reducing sugar. So which one among the following is not a reducing sugar? So obviously it is your sucrose. It's an exceptional case. Sucrose is a disaccharide or sucrose is a disaccharide, but it is not a reducing sugar. Correct? It is a non-reducing sugar. So when I say non-reducing sugar means it is not a, you can say non-reducing sugar. Why it is a non-reducing sugar? There is a reason. Because in case of sucrose, okay, you will be having a glucose and fructose linkage. The hemiacetyl carbon of glucose, that's first carbon, will be linked with the hemiketyl carbon of the fructose. That means hemiketyl carbon and hemiacetyl carbon. Hemiacetyl carbon of glucose and hemiketyl carbon of fructose will be bonded together, so they won't be free. Because to be a reducing sugar, you need to have a free aldehyde you need to have an alpha hydroxy ketone, you need to have a hemiacetal carbon, you need to have a hemiketal carbon. But in case of sucrose, okay, what happens? The carbon number 1, hemiacetal carbon of glucose, is bonded. Glycosidic bond is there between hemiacetal carbon of glucose and hemiketal carbon, that is second carbon of fructose. So they are bonded, they are not free. So they won't act as reducing sugars because their hemiketal and hemiacetal carbon are bonded in sucrose molecule. I hope you have understood. That's why it acts as non-reducing sugar. That's all for today. Meet you in the next class. Yo yo.